Before we do get started, though, did have a quick question for you. Since there are so few people here, and actually not that many have signed up, not quite sure why, would you be interested in trying to change classrooms to somewhere maybe a little closer to the other side of campus and a room that doesn't have this crazy lighting system in it? So I will make a request to do that because this is a little crazy in terms of numbers of people. It's weird. Is there another class at this time that people had thought about taking that might have been an issue? Not that. Well, I guess you're all here, so I should ask the people who are not here. Maybe that would be a better way of thinking about it. So it's fine, actually. I think it's great. The fewer people we have, the more interaction we can have. That's actually great. I wish I'd had this side classes all the time, but we shall see. So we got any lights in the back, on the side. Again, crazy lighting in this room. No, it's only these two, and these are going to be way too much, right? Okay, so sorry about everything being ridiculously dark. So today we'll continue basically where we left off last time. We talked about virus definitions, and sort of the big take-home message there, of course, is that viruses are not virions, and you're going to hear a lot about virion structure from the guy who's hiding in the back eating his breakfast right now. He's going to wave, right, Nacho? He'll be your guest lecturer on Friday. We may or may not be able to record that lecture. I'll talk to Nacho about that and see if we can, but if not, I recommend bringing like an iPhone and just doing a voice record, and I can definitely post that as well for those people who, for whatever reason, couldn't make it today. So he'll talk a lot about virions. Why should we be studying viruses? Yeah, okay, fine, they make people sick, but I'm much more interested in virus studies because they're incredibly ubiquitous. They're everywhere. They're in our genomes. They're in all environments that anybody's ever looked at. And again, podcasts I was listening to this morning, even intracellular compartments like mitochondria and chloroplasts can have viruses as well. So they're really absolutely everywhere. They outnumber all other biological particles by probably an order of magnitude, if not more than that. And they're incredibly diverse. So today, I wanted to go over how you can, first, how people discovered these viruses, and this sort of leads into some of the issues about how you can detect viruses, because not everybody has an electron microscope in their backyard or in their basement. So detecting virions is actually really pretty challenging. You can do those nucleic acid stains and see the little spots, but it's really a bit challenging. So we'll talk a little bit about the history of virology. Actually, a lot of the history of virology, and we talked about this last time, has to do with vaccines. And in fact, vaccines were developed before people really even knew about viruses and the viral nature of these particular pathogens. Because people didn't actually have electron microscopes, no way of seeing a actual virion at the time, all of the original discoveries of viruses were just due to pathogenesis. And it was literally something that was really small and pathogenic. And we'll talk a little bit about that. That's how all of these were discovered. And as I mentioned last time as well, it's only been little over 100 years since people discovered bacterial viruses. So this is a very, very recent field in terms of thinking about the most abundant particles of biology on our planet. So really fascinating question. Talk a little bit about virus detection. That will get us into plaque assays. And these are just a couple of examples of plaque assays right here. It's a way of detecting something, again, that you can't see, or at least can't see usually, again, unless you have an electron microscope. These processes, in terms of counting viruses, are really important for this concept. It's called a one-step growth curve. This was developed by a couple of researchers, actually ex-physicists, in the 1930s, 1940s, as a way for studying viruses. And it turns out to be a really, really useful way of studying virus populations, really. It's not the individual virus particle or the individual virion in one infected cell. We're getting better at doing those kinds of assays, but we're not quite there yet. So looking at populations with this one-step growth curve, and basically to understand one-step growth curves and populations, we have to do a little bit of statistics. This is the only statistics we'll do in this class. Don't be worried. The Poisson distribution. How many of you have heard about the Poisson distribution? Know about the Poisson distribution? Can do questions on the Poisson distribution? Answer all of them properly. We will go through that in a little bit more detail. But it is very important for understanding this concept of looking at populations and populations of virions versus cells. But first I wanted to talk about how viruses were discovered in the first place. This was by looking at disease, and this was not long after Koch's postulates were being developed in terms of bacterial disease. And so, yeah, how many of you had micro? Are taking micro? Did you have a lecturer yesterday? But the basic idea of Koch's postulates is that you have something that causes disease, you isolate whatever that is from a diseased person, animal, etc. You make more of that separately from everything else, and then you take that isolated, in this case, disease-causing agent, put it back into an uninfected organism and discover that it becomes sick. And so that's how people were thinking about diseases. Louis Pasteur was very active at this time as well. And they'd shown that quite a lot of diseases were in fact caused by these bacteria. And one way that you could make sure that the bacteria were being removed and not causing disease, you put them through these filters. And these filters were such that they would block any kind of bacteria. And in most cases, they would stop whatever disease that was going to be occurring. But what was weird is there were a few diseases, and these were particularly diseases of plants, where people knew about plant diseases, and particularly tobacco, which was a big deal at the time. And what they had was a disease of tobacco, and they could isolate the disease-causing agent from the tobacco. They couldn't make any more of it. They couldn't grow it separately from the tobacco, because we talked about last time, viruses need to, the virions need to get their genome into a host in order to be able to replicate some more. And that process, these things, however, went through the filter. And if we went through the filter, they could still actually cause disease. The communications at that time in the late 1800s were not great. So there are actually two separate researchers who basically were studying exactly the same thing and came up with exactly the same results and didn't really know about it. Ivanovsky in what was then Russia, then became the Soviet Union and became Russia again, and Bierjink in the Netherlands. And they basically found exactly the same thing as this small infectious agent. And so literally it went through these filters and could still cause disease. And Bierjink did a really cool set of studies looking at particularly tobacco mosaic virus. This is an electron micrograph now of tobacco mosaic virus, these long rod-shaped forms. He actually showed that these virions were very stable to drying out, to heating up, etc. He did all kinds of really interesting experiments. And he's really credited with coming up with the term virus for these kinds of infectious agents. Originally, Contagium vividum fluvium, that eventually became an ultra-filterable virus. It went through these filters, and eventually we started to call these viruses. Soon after they discovered these diseases of plants that, again, could go through these filters that blocked all bacteria, couldn't grow by themselves, the foot and mouth disease agent, the disease-causing agent here, was shown to be very similar. And the reason these guys didn't, again, fulfill Koch's postulates is you couldn't grow them in the absence of some kind of host. And so made them then, A, they were so small, they went through these filters, and B, they couldn't actually be grown. These were the first plant viruses to be discovered, first animal viruses to be discovered. And it actually took a while until, again, two independent researchers came up with studies of bacterial viruses. These were the bacteriophages. And the bacteriophages were discovered literally looking for something that would treat a disease. So they knew that there were bacterial causing diseases, in this particular case cholera and diphtheria, and they were looking for ways to treat these diseases, and they found these viruses that were able to treat the diseases. And this was Félix Terrel, and Félix Terrel, really fun story, actually big book about his history, was a French-Canadian researcher who then moved to France, and then from France moved to the Soviet Republic of Georgia, was very strongly supported by the Soviet Union right after the Bolshevik Revolution, and was really instrumental in introducing this whole idea of phage therapy, so using bacterial viruses to treat bacterial diseases. And people forgot about this for long periods of time when antibiotics were developed, but now people are coming back and looking at this. In fact, one of the leading places in the world where you can get treated for bacterial diseases with bacterial viruses is in the ex-Soviet Republic of Georgia, and the Félix Terrel Center for studying, or the Alavia Center there in Tbilisi. So this, again, little over 100 years ago, where these small infectious disease causing particles, but you had to have a particular host for them. So this sort of immediately brought up the question of, are viruses alive? And people often ask me this when I'm giving public presentations, are viruses alive? And what my immediate answer to that is to obfuscate and say, viruses are part of life. Which of course completely avoids answering the question. But it really comes down to your definition of what's alive. And if you think about viruses from the point of view of that replication cycle that we talked about last time, a virion is just the seed, the extracellular particle, part of a virus life cycle, then I would say pretty clearly these are alive. They just depend on a particular set of nutrients, which happens to be another living organism. And there are plenty of other organisms for which this is the case as well. I just got a wonderful present from one of my students who was just in South America, this little package of anti-worm for parasitic worm diseases. So they thought so much of me, they decided they would bring back a package of anti-parasitic worms. Go for 2 months to South America, what do they bring back? A little package of anti-parasitic worm disease. So, but no one, I think, would really argue that these parasites are not alive. Then why should you argue that viruses are not alive? But a lot of people do argue that they're not alive. And one of those arguments actually was, at least the virions can be crystallized. And can you crystallize life? Usually, nope, people think you can't. On the flip side, they evolve. I do quite a lot of work with NASA, and particularly from the point of view of astrobiology, so trying to go to other planets and discover if there's life there. Well, you better have a pretty good idea what life is before you go there. And one of NASA's definitions of life is being able to undergo evolution. And viruses are, I would argue, and Nacho probably would as well, that they're the experts in evolution. Probably do a better job of evolution than all cellular organisms probably combined. On the other hand, people argue that, well, you can actually write down the molecular formula, no, I don't expect you to remember this, for polio. So polio is C32,652, H1 92,000, etc. So there's a chemical formula for a virion, but that's what a poliovirus virion is. There's actually a chemical formula for that. And there's some discussions about this and also some of the biosecurity, etc. On the flip side, we're kind of bouncing back and forth here. Viruses are clearly reproducing. As long as they've got an appropriate host, they can go through their life cycle. They make more of these virions. More viruses can go on through more virus life cycles. So I would argue that viruses are alive. In the past, if you go back and look on D2L, I've actually asked a question on the problem set, which I'm not doing this year, although with 20 people we almost could. Are viruses alive? And describe why you make that decision. And no answer for that was correct. It had to do with your arguments one way or another. But again, my standard answer to that is viruses are part of life. And at least life as we know it, like us sitting around here in this room and me standing up in front, that would definitely not have existed in the absence of viruses. So talk a little bit more about some of these neat things that people can do with viruses. Part of the reason that I study viruses, the reason I started working on viruses in the first place, is that a lot of what we know about us, and really life in general, comes from the study of viruses. So classically, molecular biology, that DNA is the genetic material, comes from, well, also transformation of bacteria, but the sort of nail in the coffin to show that DNA was the genetic material, at least the cellular genetic material, came from Chase and Hershey's experiments, where they were using a bacteriophage to show that it was the DNA that was being maintained and not protein that was being maintained in terms of a genetic material. The discovery of recombination. Yeah, we always talk about meiotic recombination, where you get crossing over in terms of the chromosomes, but the first discovery and the first studies of recombination were all done in viruses, and particularly in bacterial viruses, promoters where DNA polymerases, replication DNA polymerases, but for transcription, the RNA polymerases, promoters where they sit down on DNA. All of these studies were done on viruses, etc. So this was really pioneered by, as I mentioned before, a group of, I guess, renegade physicists is one of the ways of looking at it. A lot of people say these were lots of physicists who were heavily employed in the Manhattan Project right after during the end of the Second World War, had nothing to do. And so they said, hey, biology sounds kind of cool. Let's go and look at that. And so the real originators of molecular biology were also called the phage group, and a number of, again, mostly physicists who decided they wanted to use thinking about physics in terms of trying to understand biology at a very, very fundamental level. So really the molecular aspects. The study of viruses extended from these bacteriophage biologists. People then started looking at eukaryotic viruses, and particularly tumor viruses. And we'll talk quite a bit about tumor viruses as we move on through the course, actually after the midterm. But a number of cancers were found to be formed by viruses, and you had actually had infectious cancers. Turns out this is the great minority of cancers are caused by viral infections. But if you want to study cancer and be able to say, okay, I'm going to get cancer in this particular organism at this particular time, if you've got a virus you can use to do that, it's actually a very convenient way to look at that cancer progression. And understanding how the virus is replicating and how the virus is causing cancerous development in the cell and in the organism has told us huge amounts about how cancer works, but also just in general how different aspects of eukaryotic biology work. Some of you may or may not remember from molecular biology last term when I showed the image of the eukaryotic replication fork. It's got this, at the replicative helicase, it says large T. That's the large T antigen actually from a virus. So the very first studies of looking at eukaryotic replication were actually looking at viral replication. It took much longer to figure out what the cellular proteins that were involved there. Oncogenes were originally discovered in some of these cancer viruses, and even retroviruses were originally discovered by studying cancer. A lot of when people talk about retroviruses, usually people think about HIV, but they've been studied, in fact, for quite a long time before HIV appeared. And that actually, I think, made a huge difference in terms of now having HIV treatment, is that people had studied retroviruses well before any of these immunodeficiency viruses came along. So that's not everything. Turns out you can actually, that's understanding the cells by studying the viruses. You can also now use viruses for good, and I mentioned this a little bit in the video I showed at the end of last lecture, the Edge of Life film that we're, in theory, still working on, 7 years later? Not much has happened in that period of time. But one of the favorite things I'd like to think about is this phage therapy. So again, this was developed originally by Félix Terrel, a little over 100 years ago now, where he was using bacterial viruses to treat bacterial diseases. This was completely out of favor for probably 40, 50 years, at least in the Western world. The ex-Soviet Union actually was still continuing to use this, and the Georgian, in fact, apparently I heard the Red Army would have these kits of little bacterial viruses that they would carry with them in their first aid kits that they could use then if they ever had some kind of skin wound. So those are still being used, but now people have started to use those again here, and there's a brand new group, I think they started it last year, at UC San Diego, where they're looking at trying to use these bacterial viruses for treating now antibiotic-resistant bacterial diseases. And a lot of this happened because there was I think, professor of cardiology who came down with an antibiotic-resistant infection and was in a coma, and they had no idea what to do about it, and were able to find a bacteriophage by looking all over the place. In fact, the dean of the School of Medicine, conveniently his wife, was very active in this, and they found a bacterial virus that could treat his disease, and now he's perfectly happy and fine and giving TED Talks, and they founded this whole center. So really amazing recovery. There are a few cases like that. Of course, not all of them work. They're trying to look at them in much more detail. And then there's also a really nice eye biology talk on phage therapy by a colleague of mine, Paul Turner, who talks a little bit about how that works and the kinds of things that they're doing there. Gene therapy as we know it now wouldn't exist without viral vectors. So treating genetic diseases, what do viruses do? They move genetic material around. As Stedman says, they're a bag of nucleic acids. Well, you can put whatever inside that bag you want to do, and then try and use that as a way of moving those genes around. So gene therapy as we know it, and all of the mechanisms of doing changes like CAR T cells. Any of you heard about CAR T cells? Sort of the latest immunotherapy processes. All that's dependent on use of viruses and viral vectors. To do really personalized medicine is dependent on all of these viral vectors. But it's not just medicine. Battery technology, people actually using some viruses. In fact, that tobacco mosaic virus I showed you, really long and thin, the virion, turns out that's really useful for trying to use this as a nanotechnology template and set up some batteries. Cancer therapy, so not just CAR T cells, but in fact using viruses to in fact try and treat cancers. Some of this was actually supposed to end up in our documentary, but there's a little discussion about that. And then I posted this short, I think it's like 2 pages. The Return of the Phage is actually a blog post from Small Things Considered. Elio Schechter, who's an emeritus professor, first from Tufts, but now he's at San Diego State, wrote a nice little story about talking about some of the really fun things that people can now do with bacteriophages. So big picture stuff, what you can do with viruses, how amazing they are, but first you kind of need to know how to study them. And as I said, not everybody has an electron microscope, so how do we go about studying viruses? How do you count them? How do you have a good idea how many viruses, or particularly how many virions, you have in your little test tube? I showed you the test tube last time with the 10 to the 11 PFUs, and PFUs are active virions in my little tube there. So a couple of ways of doing this. The original way was actually infecting organisms. Now, this works if you're really interested in pathogenesis. It's a great way of studying viruses, but it's really expensive and takes a very long time. And there are some ethical issues as well, particularly if it's some kind of lethal infection. But if you're interested in pathogenesis and really want to understand how many pathogenic viruses you have in your little test tube, which is clear because you can't see the individual virions, using animals is something that you really can't avoid. If you have access to an electron microscope, this is a great way of counting virions. There is ever a problem with using electron microscopy, other than the fact that the instruments cost $1 million plus usually, and training is a couple of $100s to do it, and it costs $100 an hour to use them. And then the particles that you see, sometimes your prep works, sometimes it doesn't. But there's no way of telling if you see a virion under the electron microscope whether it's infectious or not. And so it may be an infectious particle, it may be a non-infectious particle. So probably the best way to study number of infectious particles is this thing called the plaque assay. And I showed you a couple of examples before, but I'll walk through it in a lot more detail right now. So the whole idea of a plaque assay is you're looking for infectious units. And how do you see those infectious units? You basically look for some kind of infection. And the way that that works, usually you've got a very concentrated suspension, and then you do serial dilutions. I know a few of you are in the mutant virus from hell lab, so we just did those serial dilutions yesterday or the day before in the lab. 10-fold dilutions, 2-fold dilutions, you name it. And then once you've diluted this to a pretty large amount, you take an aliquot, or a small amount of this last dilution, and then you put it on a plate. Now, what do you have to do with this plate? What you do here is you mix this with a vast excess of your host cells. And the key here is that you're going to have mostly happy, uninfected, normally growing host cells here. But you'll have a few that get infected by these infectious particles. Then what you do is you let these cells grow. You'll have infected cells, which will then produce more virions, which will infect the cells around them, which will produce more virions, etc. And you'll have all the cells that are perfectly happy. Now here, in this case, the perfectly happy cells are what's in the background. So this brownish orangish stuff. And then all of these dots, the white dots on here, these represent where there was one infection that took place and has now spread and infected all the cells around that. One important aspect about this is you want the infection to stay in one place. So you also want to have some way of stopping these virions from getting all the way across your plate and infecting all the other cells. You should just infect the cells that are right around that individual one that you started with. Great thing about this is now you can just count these little dots. You count all of these dots, and then you figure out an average. You back calculate to what you had in your original dilutions, and now you get a number of plaque-forming units or infectious virus particles per milliliter that you started with here. And in this particular example, it's 1.2 times 10 to the 9 infectious particles that you had in the first place. Let's look at another example of this. This is, in fact, a plaque assay that I did when I was still in the lab and could actually do some of these things. I can escape from my office. This is the bacterial virus Phi-X174. And these are plaques right here. The little black dots are me counting each of those plaques. And in the background is the E. coli that has been infected. These are the same dilution here. We've got 67 here, 81 here. You'll notice some of these things run together, so counting them is not always completely straightforward. But this is a nice example of a plaque assay right here. How we've done that, again, we've got our virus dilution. We mix it together with the top agar, which was stopping all of these viruses from spreading too much. Here's our host. Mix these together, put them on an agar plate. Let it grow, this incubation process. So it's multiple rounds of infection here. If you're just looking at one round of infection, then you would never see that infection because it's going to be one cell. It's going to have to go through multiple rounds of infection. Each of those plaques, again, you can count all of them. This is not just true for bacteria. You can also do it for mammalian cells. This is the yellow fever vaccine. This is also a plaque assay that I did, now using viro cells, which are monkey kidney cells. Here we have one plate which has no virus on it, a 10 to the minus 1. We like to call this TMTC, too many plaques to count, TMTC. And then just through dilutions, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, you start to get a countable number, and 10 to the minus 5, you get a countable number. You got a 10 to the minus 6, there's none left. And so here, clearly, the titer is considerably lower than the titer that we had with the bacterial viruses in the first place. So this is a great way of studying virus infection, but you have to have a cell culture and a way of putting these cells onto some solid surface so you can detect them. And yeah, not all viruses you can do that. There are some other techniques that people use for testing viruses. This is now more of a clinical technique. Plaque assays, because you've got this long incubation period. Depending on the virus, this can be, I think for the FOX174, that was an overnight, but for the viro cells, it's 5 to 6 days. And if you're trying to do some kind of clinical diagnosis, this is not a really fast way to do things. So one way to do these kinds of clinical diagnoses is something called hemagglutination, and it's much, much, much faster technique. Unfortunately, this is not detecting infectious virus anymore. It's just detecting a virion which can clump red blood cells together. Now, clumping red blood cells together, why would a virion want to do that? Well, it turns out that virions, of course, need to get their genome inside a host cell in order to be able to replicate. So what do? They bind to the outside of host cells. Red blood cells have a molecule called sialic acid on the outside, and many viruses that we'll talk about through the rest of the term will have virions that have proteins on the outside that bind to sialic acid. So what happens is if you have a virion, which is shown here as the little black dots right here, little black dots will bind between all of these different red blood cells and cause them to agglutinate and basically give you this kind of amorphous mass of blood cells in the bottom of a plate. If, however, you have no virus, the red blood cells do not cross-link with each other, and they end up as a little dot right down here at the bottom of your plate. And so here's this example here. We're talking about positive controls and negative controls. The positive control, here we've done a serial dilution. Now instead of doing a serial dilution, a tenfold dilution, which is what we were doing for that last example, now we're just doing twofold dilutions. So 1 to 2, 1 to 4, 1 to 8, 1 to 16. And at these low dilutions, you notice everything is amorphous. And then once you get out to 1 to 10,000, you have this little dot at the bottom. You know the little dot at the bottom is no virus because no virus control is right here in B. Then C through H are going to be different samples, say like patient samples. And you'll see that D and E have this cross-linking, maybe H as well, whereas C, F, and G don't. This is a great, very fast, very cheap technique for doing. You can tweak it a little way. You can add different things that will block the outside of the viruses. Often people use antibodies that will block the virus interacting with these hosts, but lots of different ways of doing it. The big downside is it's very low sensitivity. We're down to 1 in 10,000 here, and there's absolutely nothing which is happening. And as you saw in the plaque assay, most of these viruses are going to be 10 to the 5, 10 to the 6, 10 to the 7, so well beyond this. I actually did my own hemagglutination assay here. This is what it starts out like, and you just let things precipitate, and what you'll see here is very few of my cases actually had anything. Right back out here, we had a little bit of agglutination at some of our lowest dilutions here, but this dot at the bottom means we have absolutely nothing here, at least of those kinds of dilutions. So these are different ways of doing things. Hemagglutination, again, fast, just gives you virus particles, doesn't tell you about infectivity. The plaque assay is really the way that tells you about infectivity. So wanted to spend a little while talking about the difference between PFUs and numbers of virions. So this is why that hemagglutination assay is not always the best, because often you'll have virus particles that are not infectious. And it turns out this varies from virus to virus. Bacteriophages, most of the time, it's almost 1-to-1, the ratio between virions and plaque-forming units. So, and when we talk about bacterial viruses in the next couple of weeks, we'll kind of see why that is. But some viruses, HIV and hepatitis B are classic examples of this. They'll have 1,000 particles and 1 PFU. So really, really inefficient processes. Now, why is that? Sometimes the particles are actually not complete. They haven't been made properly. The assembly process isn't working very well. This is true for hepatitis B. And in fact, the original hepatitis B vaccines were just purifying non-impact particles and using that as a vaccine. Worked incredibly well. But unfortunately, some of the real viruses occasionally came through there. Defective genomes. When we talk about the different viral genomes that you can find in a particular virion, some of these are, in fact, many of them, particularly we're talking about RNA viruses, have so many mutations in them or mutations in very specific parts of the genome, they're not going to work anymore. That particular virus genome is no longer functional. You can have a completely empty virion. In fact, you just call this a capsid because there's no nucleic acid left in it. And then, particularly if you're talking about whole organisms, but also some cells, if you have particular antiviral defenses in whatever system you're using, you could have a bunch of infectious virus particles, but if there's a really good protective mechanism inside the cells, you're never going to see plaques. So lots of different reasons that you could have your PFU be less than your number of virions. But all of these three top ones are these are actually not really infectious. And so that, again, is one of the issues with counting particles. And even what a lot of people do now, particularly for diagnostic purposes, is they will detect virus nucleic acid. And virus nucleic acid, you see, is evidence of viral disease. Well, that nucleic acid might not be infectious, and it might not be associated with an infectious particle. So molecular techniques are great. They're really easy. They're really quick. They're very specific, but sometimes they'll just give you the number of genomes. They won't necessarily give you the number of infectious particles. So why do we care so much about infectious particles? Mostly because if you want to study viruses, you really care about the infection cycle. So how do people study infectious cycles? The best way to do this would be to look at one virion infecting one cell and following that over time. We don't really have good techniques for this, although we're getting closer. Some single-cell analyses techniques of doing this. So you really have to look at a population of virions infecting a population of cells. And what you want to do is you really care about what's happening with that one virus in that one cell, but the only way you can look at it is an aggregate of lots of different ones. And so to do that, what you want to have is a simultaneous infection. So lots of virions infecting lots of cells at exactly the same time, and then you just follow what happens in all of those cells over time. And it turns out that viruses can replicate either in fact, in some cases less than 20 minutes. That 5x174 that I showed you actually I think can do 12 minutes from infection to producing more virus particles. But many viruses actually will take days to go through this whole cycle. This is an example here on the right from our textbook where we're looking at I think this one was SV40. Yeah, this one's a simian virus 40, where you do an infection at this point, and then you just wait over time, and you look at different aspects of this. So the first one you can look at is just the number of infectious viruses, and this you can do by your plaque assay. So this is the black line. What you'll see is it goes from a pretty high number when you start, goes down to 1 PFU per cell. We'll see why that is in just a second. And then after a certain period of time, in this case it's about 15, 16 hours, then you start to see more and more of this infectious virus and get to, at 40 hours, after about 2 days, a nice high amount, and then it actually is pretty flat after that. But what you can also look at during this simultaneous infection is everything else that's happening with this particular virus. So people are looking at viral RNA. You can look at viral RNA. You'll notice that that starts to be made well before you have infectious virus. You start to make viral proteins, in this case T antigens, well before you have virus particles. You have viral DNA, which is being made, some of the viral capsid proteins, which are made in the dashed line here, and then finally you start to make these infectious viruses. And so it's in studying all of these different phases, you can study now, and you can isolate, for instance, all of the replicating particles at a particular time in your process. So how do you do this? one of the real keys, again, is having the simultaneous infection. If you had infections that are happening at different times, there's no way you can do this kind of study because all of these things are going to be mix and match relative to each other. So you have to use what's called a high multiplicity of infection. So MOI, we'll talk about quite a bit. Multiplicity of infection is just literally number of infectious particles per number of cells. So multiplicity of infection, high, 10 is a really good one. We talk about the Poisson distribution. We'll get to that in just a second, why that's good. And then just literally follow number of particles or amounts of DNA, etc., over time. How do we do these things? Here's another example. This is now a stylized version just looking at infectious virus production. You add your virus to concentrated host cells, but now instead of in your plaque assay, remember plaque assay you use a very low amount of virus versus your host cells because you want to have those individual separated dots. Now it's the complete flip side. We want to have 10 times or 5 times a high MOI. So mix these together, and then you want to get rid of those extra virions because you don't have any more infections happening. So do this high MOI infection, get rid of the extra virions. That can be a dilution or a wash. And then just over time, do these plaque assays. And so here's just an example. Again, over time, you're not seeing any infectious virus particles. People also call this the latent period. And then you start to see infectious virus particles. We're mostly interested in what's going to go on in this latent period. Afterwards, assembly and release are interesting, and we'll talk a little bit about that. But it's really in terms of virus replication that we're mostly going to be talking about. Different enzymes making nucleic acids and making the whole protein coat. Why are these so useful? Well, it turns out that just in doing a one-step growth curve, you can get a pretty good idea, even if your virion is a naked virion or an enveloped virion, and also will tell you a lot about how these viruses are likely to be replicating. So here are a couple of examples from the Flint textbook, which I have yet to put online in the library. Sorry about that. Where you can actually do experiments with a one-step growth curve. And their classic one-step growth curve here, again, high MOI, goes to a flat line, and then goes up to a high number. In this case, we have looked at intracellular viruses versus extracellular viruses. So what you can do is, as you're doing your one-step growth curves, you can break open the cells and then see if there are any infectious virions inside the cells. Or you can wait until infectious virions show up on the outside of cells. And what this gives you is 2 different measurements in terms of time. And here, this is a nice example here of adenovirus, where we have extracellular infectious virions that only appear about 4 hours after intracellular virions appear. And what that means is this particular virus gets assembled inside the cell, and it's infectious, but it hasn't gotten outside the cell yet. So there's some extra process for getting this from inside the cell to outside the cell. And what this also tells you is that it doesn't seem to need to go out through the cellular membrane in order to become infectious. And we talked about naked virions versus enveloped virions last time. Enveloped virions have lipid on the outside. Where do they get that lipid from? They get that lipid from the cells. And it's usually in the process of coming out of the cells that you get them. So here's an example of an enveloped virus. Right here, Western equinoencephalitis virus. Here, the intracellular and the extracellular particles appear at exactly the same time, and this picks up its envelope as it's coming outside of the cell. So if you see virions that have a different intracellular versus extracellular, it's a pretty good indication that this is a non-enveloped virus, whereas if they come out at the same time, highly likely to be enveloped viruses. The other thing that you can look at here is just the absolute scale and literally the slope of each of these lines compared to bacterial doubling. These are all now on log scales. Doubling gives you a nice, even, relatively slow replication rate. These guys, instead of starting more or less at 0, they are flat for a while and then really take off. One of the things that lots of people get confused by is what is this flat line here at the beginning, and what is this PFUs per mil or PFUs per cell that starts out down here. This is, let's back all the way up to here, starts out and gets down to about 1. Now this is PFUs per cell. The way that you do these assays is you've done a plaque assay, right? So you've taken whatever you have, you do dilutions, you put it onto a lawn of non-infected cells. Well, if you have a cell which is infected that has a virus particle in it which is infectious, one of the really important things about those plaque assays is that it's multiple rounds of virus replication that happen. So if you have an infected cell, even if at that point there aren't any infectious virions in it, eventually it will produce a plaque, because in the plaque assay you go through multiple rounds of replication. So anytime you have an infected cell, you're always going to have a certain number of plaque-forming units. And so that's why you always have this low level right here. And what that means is you can look at the number of plaques that are formed during this latent period, and that will give you the number of infected cells that you have, because there are no extra virions which have been produced, because in the latent period you haven't produced any more extra new infectious virions. Then you can look at the number of infectious virions you have here at the very end and just divide this final number of virions by the initial number of virions, and you get something called the burst, which is the number of virus particles that are being produced per number of cells. So one-step growth curves, you can get these numbers, you can get an idea if they're enveloped or if they're naked, and also timing, so you can do all of your experiments looking at protein production, etc. So one-step growth curves are incredibly useful in terms of understanding a virus replication cycle, and you can learn a huge amount from studying them. And we actually try to do that with a lot of our viruses in my lab as well. So some of you may be wondering, why we have these high MOIs. Why am I talking so much about MOIs? That's because we're talking about populations. And so this brings us to the joys of math and Poisson distributions. If I were to give you a question about this on an exam, I would be sure to give you this formula and hopefully give you some relatively straightforward numbers to work with. So the basic idea here is that if you look at, this is their standard Poisson distribution, but we're interested in the number of cells, or literally actually it's the fraction of cells that are infected by K number of particles, in this case virions. So fraction of infected cells. What we're trying to do for the one-step growth curve is have P of K be as close to 1 as possible. We want that to be something which is near to 100%. But we're talking about populations here. And so even if you have 5 cells and 5 viruses, they're not always going to end up with each of those cells being infected. And that's described by this process. This formula, I should say, of the Poisson distribution. E to the minus m. M is your MOI, number of virus particles divided by number of cells. M to the k, which is this proportion that you have here, divided by k factorial. Now, why is this? I made a little animation to sort of show this. At an MOI of 1, 5 virus particles, 5 cells, this is, again, your random distribution. Sometimes you're going to have cells that have 2, and sometimes you'll have cells that have none. If you do an MOI of 2, you'll have more likely, but you'll have some cells that have 1 and some cells that have 3 associated with them. An MOI of 5, it's highly likely you're going to have multiple virions associated with multiple different cells. And again, this is really well described by a Poisson distribution. Here it's the A rather than K. But if you look at number of, so A here is the number they're infected by a particular number of particles. At an MOI of 2, so 2 virus particles, how many cells are you going to have that have 0 virus particles that are associated with them? Turns out you get about 13%. Even if you've got twice as many virus particles as you have cells, about 13% of them are uninfected. Is that going to be any good for a simultaneous infection? Even at an MOI of 3, you still have a pretty reasonable, almost 5%, that are uninfected. It's only when you start to get to MOIs of 5 to 10 that you get this proportion of uninfected cells to a relatively low number. And we can look at that here. What we really care about is going to be P of 0 or P of 1. So how many have no virus particles associated with them, and how many have at least one that's associated with them? So P of 0, you just, again, use our formula down here. 0 factorial is 1. M to the 0 is also going to be 1, so it's e to the minus m is the proportion that you have at 0. So at an MOI of 1, 36% of your, actually 37% of your cells are uninfected. An MOI of 2, 13.5, an MOI of 5, 0.7, and an MOI of 10. So why don't you always do an MOI of 10? Because often you don't have enough virus particles to do an MOI of 10 if you're doing a one-step growth curve. Usually it's going to be this balance between having enough viruses and enough cells to be able to do the infections with. So the number of uninfected cells, this is bad, but then what you want to have is what's the difference between your infected cells, or more than one. So since this is a proportion, your 1 minus P of 0 is P of 1, P of 2, P of 3, P of 4, P of 5, P of 6, P of 7, P of 8, etc. Out to infinity. So 1 minus P0 is going to be your number that are infected by more than one of these infectious particles. MOI of 1, 63% infected. MOI of 10, 99.995% infected. So people get this. This makes sense? Yes? No? Huh? Okay. We could do this clicker question. Do we want to do a clicker question? Or should we move on and talk about classification purposes? I'll let you just do this by yourselves. So what do we talk about today? Where do viruses come from? Again, these are those small infectious particles. Are they alive? They're part of life. I would like to say that they are, but we can think about that. All kinds of really fascinating things that you can do with viruses. Most important, as far as we're concerned, these plaque assays, one-step growth curves, and the Poisson distribution. So in the last 10 minutes or so, I want to talk about the interesting stuff here, which is basically everything that happens between the virion binding to the cell and more virions going and infecting the next cell. So it's this process right here that we're going to spend the rest of the time in the course talking about, and the differences and similarities between the different viruses in all of these processes. How they bind to the outside of the cell, how the genome gets outside of the virion, how these genomes get transformed into proteins, how these genomes get transformed into more genomes, how these end up making the proteins which are important for the virion structure, and then how the virion gets to the outside of the cell. So we'll spend a long time talking about this, again, for each and every virus as we move through this later. But before we do that, I wanted to talk about a little bit about some of the Baltimore classifications, actually slightly out of order here. So what is the process? And this is, again, overall, in each of the different viruses we'll talk about each of these steps separately. How do virions bind to cells? They bind to basically anything that's on the outside of a cell. They can bind to proteins, they can bind to lipids, they can bind to carbohydrates. You name it, there are viruses that are going to associate with something on the outside of the cell. And what cells and what particular cells virions bind to is defined as tropism. So what cells can these viruses associate with? How does this genome get released? Lots of different ways. My favorite is the drill structure, which some bacteriophage use to literally drill through the outside of the bacteria. Sometimes the virions can get in through damage to cells. It's very true for plant viruses. You can have fusion between either the envelope of the virus or between multiple different cells, or the capsid can get broken down. May have noticed here this early gene expression versus late gene expression. So early gene expression, we'll talk about this again for lots of different viruses, but usually early genes are those genes which you need to make more genome, and late genes are what you need to make more virions. And so we'll talk about the different ones a lot more later on. So the early genes are almost always going to be genome replication. The late messenger RNAs, transcriptions here, almost always happens after you've made more genomes. And this is not surprising because most of the genes that you need to make virions, and Nacho will talk a lot about this on Friday, you need many, many copies of those genes. You need a lot of the virion proteins. And so you need many, many more of these, and the way to get many, many more of these is to have lots of copies of your genome first before you start to make them. What are these genes? They can be actual structural proteins, but often there'll be scaffolding proteins that we'll talk about that help form the structure, and budding proteins that will also help, again, put the structure together. How do virions get out? Lysis, by blasting open the cell. This is sort of the most spectacular process by how you get more virions. They can literally just bud off of the cell, or you can have cell-to-cell contact. You can have something which eats something in the cell. Classic example, of course, here would be the mosquitoes, which are moving things around, etc. So again, we'll talk about this for all of these different viruses. This is all that's happening in this latent period. As I said, this is the important and interesting period from the virus replication point of view. Not so much for any of the rest of these. So finish up talking about Baltimore classes. People love to, yeah, sure. So lysis is happening, well, it depends, but here's the external form of the virions. They're actually being produced right here. And so lysis will start to happen right here. The other questions? Sorry on this stuff. I'm going pretty quickly. Okay, so the Baltimore classes. We have a separate way of being shown in our textbook, but this lots of different ways of thinking about the Baltimore classes. I'm not going to ask you which, what's Baltimore class 6, what's Baltimore class 3, what's Baltimore class 1. I can't remember these things. But the key to the classification of different viruses, through this Baltimore classification, really has to do with, fortunately where my pointer is right now, about the messenger RNA. How you get to messenger RNA. Because we mentioned last time, all viruses are dependent on cellular translation machinery. So it's always got to be the virus that makes that RNA, or gets the RNA made, and then it's cellular machinery. That's what this arrow is over here. That's all cellular machinery. But how you get to this messenger RNA happens to be different in lots of different kinds of viruses. And this is how David Baltimore decided to classify viruses, other than viruses that make different kinds of organisms sick. And we'll use this classification, almost all virologists do, for talking about different virus classes. So we'll talk about DNA viruses, we'll talk about RNA viruses, we'll talk about retroviruses. And that's the kind of genome that's inside the capsid. So Baltimore class 1, these are double-stranded DNA viruses, get transcribed to messenger RNA. You've got single-stranded DNA viruses, those get made into double-stranded DNA viruses. And then you've got all the RNA viruses. We've got positive strand RNA viruses. This actually can be used as messenger RNA. That's what this plus means. The plus means it's actually coding. But to make more genome, it's got to go through a negative strand RNA stage, and that then becomes your messenger RNA. There are some viruses that encapsulate negative strand RNA. That gets copied into messenger RNA, and then it gets back copied to make these. There are double-stranded RNA viruses that then can copy the negative strand to make your messenger RNA. And then these really bizarre ones, and this is why David Baltimore came up with this classification, because these didn't fit with any of the other ones. There were viruses that had positive strand RNAs in their genome, but they had to go through DNA intermediates, and these are the retroviruses in order to make the DNA. And I've made a couple of little cartoons about this. So a Baltimore class 1, double-stranded DNA that then goes to your messenger RNA. Class 2, single-stranded DNA in the capsid. That ends up getting replicated into double-stranded DNA, from which you get your messenger RNA. The double-stranded RNAs can take one of those RNAs and make it into messenger RNA. The positive strand RNA viruses will make a negative strand of the RNA that then gets made into your messenger RNA. The negative strand RNA viruses will make the positive strand directly from them, and the retroviruses first make a single strand of DNA. That single strand of DNA becomes double-stranded DNA, and that then gets made into messenger RNA. And so these are all of these different classes, and we'll talk about each of these later on and literally separate each of the different viruses. Class 1s we're going to spend a lot of time talking about because these are the bacterial viruses. The vast majority of bacterial viruses are double-stranded DNA viruses. A lot of the viruses which infect plants are these positive strand RNA viruses. The big nasty viruses that are infecting humans, Ebola, etc. These are negative strand. Flu is another example of this. And then, of course, the retroviruses up here. So we will do one quick clicker thing just to make sure that the clickers are working. Which of these Baltimore classes, again, this is not going to finally count. Let's get it to actually work. Will it work? No. Yes, this is correct. OK. Which of the Baltimore classes of viruses do not use RNA as an intermediate in genome replication? Single-stranded DNA, double-stranded RNA, single-stranded RNA, single-stranded RNA, or retroviruses? And this is to basically make sure your clicker works. And I'll post these just so you can see them on D2L. So just 16 of us here? I think there are more than 16. Everyone have clickers? If you, again, I think I mentioned last time, if people don't have clickers and are having trouble getting clickers, come either go and take them out from the library if you've forgotten them on one day. But if you have a real problem with getting them for the rest of the term, come and find me. I may be able to scrummage up a clicker or two for you. OK, so what are people thinking? Do not use RNA as an intermediate in genome replication. Oh, hang on. It's, I can't even see my own clicker up here. So the single-stranded DNA viruses, or any of these RNA viruses, or the retroviruses, the genome replication for retroviruses, that genome is actually RNA. So I would say it's going to be your single-stranded DNA viruses rather than the other ones. And we could also talk about this class as being kind of a class in RNA biology, because the vast majority of these viruses, particularly all of the interesting ones, are going to be using RNA as part of their replication machinery. And so this is where you've got lots of differences relative to all of the other viruses that we've talked about so far. So on Friday, we'll have a guest lecture. The guest lecturer is sitting in the back there. And we may or may not have a chance to record it. So if someone can bring a voice recorder and then post it, I can also post that. I will also post Nacho's slides sometime before lecture. So I may or may not be here on Friday. If I am, I will see you then.